excuse me, I think we've sorted out the technical difficulties so we can make a move. So it's my great pleasure to welcome John Marioni back, actually here for the second time in less than a month, this time in 3D instead of 2D, which is nice. So John is Senior Vice President and Head of Computation at Genentech Research. Now, 6 months ago, he was doing something completely different. So I've asked him to say a few words about what he's now doing and why he's now doing it and so on, so that we'll learn something about Genentech. And then he's going to talk about single cell technologies and so on. So welcome, John. It's great to have you back.
Thank you, Simon. It's always fun to come to New York. This microphone. We thought about having Simon try and introduce me from the microphone whilst it was attached to my jumper, and decided that that would probably look inappropriate. So I decided not to do that in the end. Okay, good. Excellent. So yes, so thank you all for being here, both physical, corporal form and virtual form. So yeah, Simon says I started at Genentech fairly recently, so 7 months ago. And prior to that, I ran a lab in the UK at the European Bioinformatics Institute at the University of Cambridge for 12 years. The focus of my lab was, and still is, my research interest on cell-fate decisions, single cell technologies as a driver, and developing the statistical methodology. But as Simon said, it's in September last year, I moved to Genentech, and it's just worth giving a little bit of context on what I'm doing there and why, since it's still not so common a transition for an academic to make from academia to industry, and just to give a little bit of context. So in essence, I was very happy running my lab in the UK. I had a great bunch of people. It was a great institute to be at. I was latterly the head of research, so looking after all of the research groups at the EBI, which is a real privilege. It's for those of you who don't know, the European Bioinformatics Institute is one of Europe's, or arguably Europe's, largest center for computational biology. And it's a fantastic place to do research. You might be, for those of you who are maybe not aware of some of the research groups, you'll almost certainly be aware of things like Ensemble, or indeed the AlphaFold database. So the work from DeepMind is all hosted at EBI and then shared with the wider scientific community from there. So it's a tremendous place to be. And so why move? Well, I think we're at the cusp of an extremely exciting time in the biological sciences, and the impact of computation, the type of work that everyone in this room does, is just going to have increasing impacts over the next decade. And I think the way that we do experiments, both in academia and in industry, is going to really alter, and the role of in silico approaches is going to come more and more to the forefront. What do I mean by that? Well, I mean that we've got an opportunity, given advances in computation, both methodology and in computing itself and the hardware, so driven by amazing technologies by companies like NVIDIA and others that really are at the boundaries of what you can do computationally. We can now begin to simulate incredibly complex and large systems. This is easier in the fields of physics and chemistry, where there are fundamental rules that kind of motivate the simulations that you're going to do, but increasingly in the biological sciences, building on the data that we've collected, some first approximation to biological models that are not going to be perfect, but they're going to be hopefully decent enough. You could begin to think about how you would simulate incredibly high parameter space, parameterized by billions of parameters, and then in silico model perturbations. And by so doing, you could then think about how that will allow you to design the next suite of experiments, how you can really iterate very rapidly. But instead of starting from maybe a biological hypothesis and a data set, you'll start from a really very extensive in silico screen. And then there'll be this very virtuous lab in a loop where you generate data, you generate data in silico, you generate a hypothesis, it's validated, you have a very fast cycle. You could think of that in basic target discovery, but also clinical trials. How can we more rapidly update sampling schema for clinical trials? How can we identify the right patients? There's this notion of digital twins, where you model a human and then you kind of ask, what's the impact of a perturbation, so a drug, on them? How are they going to respond? And can you use this to understand better mechanism? And I think that it's hard in academia to have that end-to-end view of the whole process from the early research through to the clinical trials and the associated data. It's possible, but hard. And for me, given these academic opportunities, these intellectual scientific opportunities, coupled with the data that's available at Genentech and the very talented basic researchers they have there in the biological and clinical domain, it felt like a really natural time to make that transition for me, because I think that doing that at scale requires some of the resources that a company like Genentech can provide. And certainly if you really want to push these things through to designing better drugs, ultimately, you kind of have to be somewhere like that. So to give a more concrete example, in this state here in New York, out of NYU a few years ago, a company called Prescient was formed by Rich Bonneau and Kyung-Yun Cho and one of their postdocs, Vladimir. And what that company does, and they are now part of Genentech, was to think about how you could design better molecules. So we have a lot of information about large and small molecules, so antibodies that are targeting particular genes, and we have information about their affinity, we have information about their structure, and their structure could just be represented as a graph, right? And so you've got information about affinity, you've got information about other properties, you can represent the whole corpus of molecules as a graph. And what you want to do is search that molecular space to find different classes of antibodies or molecules that have not previously been designed to target to focus on particular targets. So that's one example, but there are so many others. And for me, the opportunity to be part of that, and my job in Genentech is to set up a fairly large computational organization that encompasses everything from data management through to research into clinical trials, from early research through to early clinical trials. That opportunity was just really tempting. And I think that over the next few years, the boundaries between academic research and industrial research are going to become more and more porous. There's going to be more and more swapping from one to the other. I think that's for a few reasons. I think that when I finished my postdoc, I didn't consider industry at all. I didn't think it was a very interesting thing to do. I thought I would end up just pressing the GWAS button or something like that, something very mechanical and not very exciting. But I think that industry has recognized that in order to recruit people from a more academic background, they've got to give opportunities to do research. So at Genentech, you can have your own postdocs who do anything. They don't have to do pipeline-related projects. And other companies think similarly in terms of the way that they can motivate people to do work. And at the same time, I think we all know that academia there are many brilliant features of academia, but it's also become harder in some ways in the last decade with sort of reporting structures, grant funding, constraints, etc. So I can imagine in the next decade, there'll be much more porous interchange. And this is good. It's not a bad thing at all. We should be comfortable, people going one way and then another, kind of bringing different views. So that's all I really want to say about that and why I made the transition. It sounds rather more dramatic than what it is, because on a day-to-day basis, I really do pretty much what I did before, just science, thinking about how you are going to uncover targets, how you're going to model data. But Simon thought it would be interesting. So that's all I want to say on that. So before moving on, though, if there are any particular questions on this, and then I'll get on to what is work based around my lab in the UK still, I'm happy to stop if there are any questions specifically about Genentech and that type of part of the world. Nope. In which case, on with the science. You're all totally committed to the academic model, I see. Excellent. Okay, good. Right. Okay, this is going to be interesting. Okay, I'm going to use the mouse to scroll here. Okay, so what do I do as a lab? Or has my lab done for the last 12 years? Well, I think we've used computational models to try and understand the molecular mechanisms that underpin cell-fate decisions in a variety of contexts, from early development to disease, with a particular focus on early development. I think that the lab itself has a variety of people, from the more statistical to the more experimental side, and I've always found that really useful because it kind of gives a broad range of perspectives on science and motivates the methods we develop. And of course, I hardly need to say to this audience, or indeed almost any audience in the world now, that our ability to understand cell-fate decisions has been really transformed by technical advances, especially in the field of single-cell genomics, where we can evidently sample millions of cells and characterize their molecular profiles in a variety of ways at the transcriptomic, chromatin, protein, spatial level, you name it. You can pretty much begin to think of doing it now with single-cell genomics. And that, since cell-fate decisions are fundamentally made at a single-cell level, this is a really important technology for understanding the mechanisms by which they work. So over the years, as I mentioned, we've been motivated a lot by, we just hope that you can see enough of it, by understanding this in the context, or applying these approaches in the context of early mammalian development. And the system that I'll talk most about today, so I just want to introduce it briefly, since many of you will not be probably familiar with it, is gastrulation. So this is an absolutely key point in development when you move from having a fairly homogeneous population of cells in an entity known as the primitive streak and the epiblast, and it diversifies into the three germ layers, so the mesoderm, the endoderm, and the ectoderm. And in the mouse, this happens over a period of 48 hours. So you go from something that looks like a fairly homogeneous mass, as you can see in the middle here, this is the epiblast, so just a bunch of cells that look under the microscope very similar. And within 2 days, you've begun to form a structure that resembles the adult animal with the head, the tail, and this is something called the primitive streak. So this is basically a part of the embryo where as cells pass through this structure, they begin to commit to different lineages. It's a system that also leads to rapid expansion in cell numbers. So you go from about 650 cells to about 60,000 cells over a 2-day window. And so it's a system that's really very, very suitable for single cell approaches. And so that's when we were deciding what to work on with colleagues in Cambridge. And this was around 2014. We aligned on this as just a very appropriate system for benchmarking and testing single cell tools. Okay, so just in a little bit more detail, though it doesn't matter too much, what happens here if we focus on E6.5, this is embryonic day 6.5. So this is 6.5 days after fertilization of the oocyte. You've got this structure here. This is kind of a cartoon of the embryo, and in green is the epiblast. And then over this 2-day window, what happens is that cells will pass through a structure called the primitive streak. And as they pass through the streak here, they'll receive signals, and that will commit these cells to different downstream lineages. That's where you end up with the different germ layers. So it's a really very rapid process. And so over the years, we've used an array of technologies to look at this, primarily the workhorse being, as for many people now and still the most commonly used, 10x genomics. So together with colleagues in Bertie Gutkin's lab, we looked at these 9 stages of development from E6.5 to E8.5. So this is relatively old work. This is around 2019 when this was completed, 150,000 cells, and then used these to think about how we could construct a relevant atlas. And actually collecting these data led to one of the motivation for one of the first, but a fairly early method that we thought about for combining different 10x genomic data sets. And that was how we would do this over these different time points. Because if you go back a slide, you can imagine that it's not possible to randomize these samples so that we collect them on the same day. Just technically, that's not possible. So we've got these 9 time points, and they are very confounded with batch effects and other technical features. And so then a postdoc, Lally, who's now a group leader in Berlin, came up with this approach around the concept of mutual nearest neighbors, which is quite an old approach, but one that still, I think, forms the baseline for many technologies that are applied now. So they've kind of riffed off this. It's conceptually very simple. You got 2 batches, batch 1, batch 2, 3 populations, red, blue, purple, red, blue, gold. And the very simple idea is you just, between the 2 batches, you look at the distance between pairs of cells, and then you take a cell in batch 1, look at which set of cells it's closest to in the second batch in the cosine normalized space, or whatever space you want really. And then you do the same in the opposite direction. So you ask for each cell in batch 2, which cell is it closest to in the first batch. And then if they are mutually closest to one another, you define them as a pair of mutual nearest neighbors. So these are pairs of cells that are mutually close to one another in each pair of dimensions. So just to give you an example of a pair of cells that would not be, this purple cell and this red cell here, they might be closest in this direction, but not in that direction. So they're not mutual nearest neighbors. Then having identified these, you can just compute a locally smoothed correction vector and basically collapse these populations on top of one another. And this is a way of integrating data, and it's sort of been employed, or related approaches have been employed by other technologies using more sophisticated approaches since, but it's still an approach, I think, this concept of mutual nearest neighbors that has kind of motivated development in the field. So I just wanted to briefly shout out to it here. Using that, we were then able to collapse these 9 days. You get these UMAP representations, which we're all familiar with how the data looks. So colored by different clusters, so different cell types, from early development here in the epiblast through to the blood, through to the endoderm and the gut tube, with contributions from the extraembryonic endoderm down at the bottom, and a morass, a mix of populations from neural and mesodermal cell types in the middle. So this is all now fairly standard. It still takes a lot of time, and in particular, what still takes a lot of time is this annotation. There are some automated approaches to annotation. They're still not great, and one of the things that I think is particularly challenging is that different studies use slightly different nomenclature. So as a field, we haven't settled very well on cell type ontologies. It's dry and it's rather tedious, but we were talking with some of the students about difficulties in integrating data sets and combining different data sets. If we can't settle on cell type labels, then it becomes extremely difficult, because you're kind of guessing which cell types are actually linked across data sets. So as a field, we've made great strides in a lot of the analysis, but some of the underpinnings of this, like ontologies, really we're not where we should be as a field, I think. So these sorts of things hinder our ability to really do the analyses we would want to do moving forward. Okay, then yes, we have this beautiful diversification of cell types in the ribbon plot. You can sort of see you go from a fairly homogeneous group of populations early on to a much more diverse set at 8.5, with many of the cell types that make up the adult animal. And then you can use this is just an illustration, of course, to identify new marker genes along each trajectory and for regulatory relationships. You can do all sorts of things with these data. And this was published a few years ago, so I don't want to dwell on it. It's just to give kind of an overview of some of the work we've done in this field and how we've used this to construct these atlases of development. Okay, I just want to also touch briefly, because I think it's still nice and illustrates a little bit more about some of the methods, and then I'll move on to new work, how we've used this to think about understanding regulation. And so again, as part of this Wellcome Trust Funded Consortium, this time now with the lab of Wolf Reich, who now leads the Altos Cambridge Institute, but at the time was a group leader at the Babraham. We also applied multi-omics approaches to the same stages of development. And this is a technology the Wolf's lab developed, where they could profile chromatin accessibility at methylation and RNA expression in the same cell. So you're all quite familiar with the single cell attack multi-omic approach. This is similar to that, but you also have DNA methylation as well as part of the same game. And the goal here was to study the role of the epigenome during this process. So this is the data, or a snapshot of the data on the right-hand side. This is not a very high throughput approach, so the number of cells we can profile is relatively low, just at 4 different time points. But the key thing is that in each of these different layers, the RNA, the methylation, and the chromatin accessibility, you've got the same cell. So you're able to measure multiple molecular modalities on one cell, which is, of course, what you want to do. Now, how do you analyze this? Well, there are a variety of different approaches. The approach that we've taken, and this is work with my colleague, Ollie Stiegel, who was group leader at EBI for some of the time that I was there. And this is using a multi-view factor analysis effectively to decompose these different data sets. So in a typical factor analysis, what you'll have is a particular matrix, and you want to decompose that into a series of factors, and those are going to be decomposed by, then you've got a cell matrix as well. So, and the things you really care about are the factors in the cell. So this is just analogous to something like PCA or any other factor analysis type approach. In a multi-view factor analysis, what you've got are these different views, and what you want to do is you want to decompose the data into factors, and these factors can either be common to one of the views or potentially shared across the views that they're shared. So basically we can decompose the full data set into a series of factors, and then the weights on those factors will tell you whether they're common or present in one of the views, say the expression view only, or across multiple of the views. It's relatively slow as an approach, but it's still, I think, relatively effective. I think that this is still an area that probably motivates a little bit more work to do really well, in particular, incorporating the true underlying distribution of the data, especially if you add more complex modalities. It's still not optimal, and there's a fair few assumptions here. But nevertheless, when you apply it to this data set, you're able to identify a series of factors. So, for example, the first factor explains variation in the RNA expression and accessibility view. So that's not terribly surprising, perhaps. You'd expect gene expression to be correlated with chromatin accessibility. And then you can use these to visualize the data and find sub-features. What I think was the most interesting thing, though, was that we were able to identify, which is not surprising, the demethylation and accessibility of enhancers defines lineages. I don't think it's shocking at all. This is much more likely to define lineages than what goes on at promoters, which is a downstream consequence of changes in methylation and accessibility in enhancers. What was more interesting was how these patterns arise in time. And so independently, we used ChIP-seq of HCK27 acetylation. So that's a marker of enhancers. And we did this in each of the 3 different germ layers at E7.5. So for the ectoderm, the endoderm, and the mesoderm separately. And then at the enhancers that were defined in each specific germ layer, we used our single cell data to look at the changes in accessibility and methylation profiles. So what you see here are cells that are at the RNA level are annotated as ectoderm. And then below that, we're looking at ectoderm, endoderm, and mesoderm specific enhancers defined for the ChIP data set. And that's at the methylation in red and accessibility in blue. So you can see for cells that are assigned at the RNA level an ectoderm identity, the relevant enhancers are accessible. You can see the peak there, and demethylated, so they're used. And the endoderm and mesoderm enhancers are repressed. So basically they're not open and they're methylated, so they're not used. Now, if we go down to the cells annotated at the RNA level as either mesoderm or endoderm, you see something that's similar, but actually a little bit different. So in the mesoderm cells, you see that the enhancers are, the mesoderm enhancers as defined from the ChIP-seq are indeed demethylated and accessible, but so are the ectoderm enhancers, and that pattern also holds in cells annotated or assigned an endodermal identity. And if we go further back in time, curiously, if we go to E6.5 or E5.5, what we saw was that ectoderm enhancers, and remember these are defined as ectoderm enhancers at E7.5 only, they're not defined at these earlier time points, they are actually still accessible and demethylated at the earlier time points. And you can even see hints of this going back to E4.5, whereas the endoderm and the mesoderm enhancers are closed, the chromatin is closed, and they're demethylated. So they're not used and they're not accessible. And this suggested a hierarchical model where the epigenome really would specify which cells are going to commit to different germ layers. So basically, the hypothesis is that the ectoderm-specific enhancers are marked. We don't quite know how yet at E4.5, and they don't undergo the same global repression as endoderm and mesoderm-specific enhancers, which are silenced during the global silencing of chromatin that occurs through this developmental process from E4.5 to 6.5, with instead the mesoderm and endoderm enhancers having to be actively de-repressed at later stages of development. So you've got a model where the default, if you will, is towards the ectoderm, and then you have to make an active choice to go to one of the other 2 germ layers. For those of you who work with mouse embryonic stem cells, this should be familiar, because if you just take them and let them differentiate, by default, they will be inclined to go towards a neuronal and ectodermal-like state. So this is actually consistent with in vitro experiments, but suggests, and it's still, we're continuing to work with Wolf on understanding exactly how this is done using gastroloids and quite large throughput screens to target these individual enhancers. But we don't quite understand how they are protected through this process, but they are somehow identified and protected so that they don't get silenced, unlike the endoderm and mesoderm specific enhancers. So this is work that was led by my PhD student, Ricard Archelaguet, who's also developed the methods with Ollie. So he was a joint student with Ollie, and he's now actually working with Wolf at Altos. Okay, so that's relatively old work. I'd now like to turn to more modern work over the last year or 2, including a couple of unpublished vignettes. And the first thing I just want to talk about briefly and this is something that I mentioned at Simon's Symposium, so apologies to those of you who heard it previously, you're going to hear it again, is how we use reference atlases. Also, apologies to the students then who heard about the Milo approach earlier last week, so they're going to hear about it again as well, but whatever. I apologize for that. So basically, if you think about how we use reference atlases, there are a number of ways that you can use them. You can typically what you do is you take a cell type, you would then, and in a perturbed condition, ask, how does it change? Is it present? Is it absent? And does the expression of cells within that cell type change? The trouble is that relies upon the definition of cell type, which is a very coarse thing to do. It depends a little bit on what resolution you want, and there's no solution to what is the right resolution for clustering. And so we wanted to come up with a different approach that didn't rely upon this. We wanted something that didn't depend upon cluster identity. It was applicable to data sets with different technologies. We wanted to be flexible to more complex experimental design, and we wanted to model variability in cell numbers between biological samples. And so this motivated my student, Emma, to come up with the approach that we termed Milo in the end. And what's the goal here? Well, it's a very simple approach in principle, at least. We just build a k-nearest neighbor graph, and then using an approach that was pioneered from Dana Pear's lab, we identified index cells indicated by these different asterisks here that are basically randomly distributed across the graph. And then around each of these index cells, we count how many cells are from condition A or condition B. So these index cells are defined agnostic to cluster identity. So you've just got a KNN graph. We want to distribute our index cells randomly, basically around that graph, and identify a whole series of neighborhoods that are indexed by the little asterisk here. And then this allows us just to build a very simple matrix where we've got our two conditions, purple and white, and we can ask in each neighborhood how many cells are from sample 1 from the purple condition, sample 1 from the white condition, and we can do that for all of the neighborhoods and just build a very simple count matrix. And of course, this is familiar to many of us from a variety of different settings, including differential expression. So built upon that, you can think about simply applying generalized linear models to these data here. So what you do is if they model these counts as negative binomially distributed, you test for differential abundance. That in a GLM framework that allows confounding factors to be incorporated. So if you had a continuous process, you could incorporate time here, sex, age, etc. Can all be easily incorporated. I think that one of the things that was a little bit of a tweak is that these neighborhoods overlap with one another. And so you've got to be a bit careful about independence and multiple testing correction. So we control for this by using a spatial false discovery rate. Basically, it's a weighted false discovery rate. So depending upon the extent of overlap, that influences the weights that you use in the FDR correction. So it basically allows you to incorporate any degree of overlap or the extent of overlap between the neighborhoods. So it kind of gives you a slightly more robust test. I mean, more generally, as an aside, I think that there are approaches that try and take account of correlation in testing for differential testing correction. They've, for some reason, never really been adopted very much, which is a bit surprising because if you think about genes, they're clearly correlated. They fall into regulatory groups. They are co-regulated. These are not independent tests. We shouldn't think of them as independent tests. And so the kind of vanilla multiple testing correction approaches basically assume that if this were a gene expression matrix, that all of the genes are independent. It's not true. And so that actually, you're getting a slightly skewed estimate of the multiple testing corrected p-values or FDRs if you don't account for the fact that the genes, or in this case, neighborhoods, depend upon one another. It's slightly surprising to me that that hasn't, there is one approach from Gordon Smythe's lab that kind of tries to account for this, but it's never really caught on. I'm a little bit surprised by that because it feels like a natural thing to account for. The fact, though, that it's a complicated model, I suppose, but it's because there's multiple regulatory networks affecting the same sets of genes, so it's difficult. But nevertheless, what we're doing is quite simplistic in the field, I think. Anyway, so how did we test this? Well, simple simulations, condition A, condition B. It's a simple little model here where you can see in this particular region here, we have an abundance of cells from condition B over condition A, so there's more density of cells here. And then we can represent the neighborhoods as circles, with the area being proportional to the neighborhood size. And this is the log fold change from the test statistic from running Milo. And you can see indeed that this region here seems to be popping out as one that's got a significant log fold change. So also confirmed with the spatial FDR corrected values on the top right. So it works reasonably well. I won't talk about that for time. And then we can apply this in a variety of contexts. This is a real data set looking at cirrhosis of the liver. So we've got single cell RNA-seq on the left. We've got injured and uninjured samples here. In the original study, they clustered all of these immune cells as to one group. Actually, you can see when you just generate the neighborhoods that there's definitely more structure here, with this particular region being more abundant in the uninjured than in the injured samples, and the opposite pattern over here. So you can identify more structure in the data by applying this approach that doesn't rely upon these coarse cluster labels. So that basically is what we did in Milo. And then turning now to unpublished work, I just want to touch briefly on a different application, and that's using it to do cross-species comparisons, now focusing on the rabbit. So for many applications, the mouse is really good, but actually in some contexts in development, it's different. So the sort of the shape of the embryo, the mouse is a cup, the rabbit is a disc, which is more like the human context. So, and also the rabbit is very widely used in a variety of settings as a model organism, especially in pharmaceuticals and in other contexts there. So we were interested that this predates any discussion with Genentech, actually. So that's a pure coincidence. And this work is a collaboration with Novo Nordisk, actually. We were interested together with Bertie again in generating an atlas of development in the rabbit. So what you could see in this table on the left-hand side here are rough matching of days of development between the rabbit, mouse, macaque, and human. And you can see that if you recall the mouse atlas I talked about earlier, it was focusing on Carnegie stages 6 to 9. And so what we did was we generated data from the rabbit at 3 stages, at gestational day 6, 7, and 8, that basically overlap with this window here. So the first thing that we did with this was just very analogous to what we did with the mouse data earlier, generated a single cell atlas, again with the cell types, using not only the same labels, but even the same colors here to try and make it more visually comparable anyway. This is just a UMAP. You're not meant to be able to interpret too much from this, of course, but you can nevertheless see that we're getting quite coherent groups of clusters. And if you look at the names, they're roughly equivalent to those that we saw in the mouse earlier. So it does seem to overlap pretty well. But what we were especially interested here was comparing developmental trajectories between the two species. So does development proceed the same way for particular subpopulations of mesodermal cells in the mouse and the rabbit? And can we then argue where there are differences? And so to get at this, we again used Milo. So we didn't want to rely upon annotations. Instead, what we did was in each of the two species, in rabbit and the mouse, we defined the neighborhoods in the KNN graph independently generated in each species. And then between each neighborhood, pairs of neighborhoods across all the species, we computed just a correlation matrix. So basically, or a measure of similarity between expression profiles of all cells in a particular neighborhood between species. So what you end up with is a graph with neighborhoods from one species on the x-axis, y, and then the neighborhoods from the other species on the columns, and just a whole bunch of similarities. So basically correlations between pairs of neighborhoods. And that allowed us to compare and identify similar neighborhoods in an unbiased way in each species. And then having done that, you can plot these across all regions. So what we're looking at here is a set of correlations from lowest in dark colors to highest in yellow. And so this is now plotted on the rabbit UMAP. And this is looking at the correlation between particular regions across species. And you can immediately see that the extra embryonic cells down at the bottom here are less strongly correlated between species. This isn't so surprising due to the way that implantation occurs. So in the rabbit, implantation occurs during gastrulation. So the process is actually concurrent, whereas in the mouse, implantation takes place much earlier. So you would expect there to be some difference here. Also, these extra embryonic cell types tend to be the most evolutionarily diverged. So it's kind of what you would expect, whereas the more core embryonic cell types here are actually more conserved across species. You can visualize this in a variety of different ways. So what I'm plotting here are cell type labels on the rows, and this is the distribution of correlations between pairs of neighborhoods where we just assign the neighborhood a cell type identity based on the majority cell type label within that neighborhood. So you can see that down at the bottom, if you look at the labels here, those in orange are extra embryonic, those in black are embryonic, and you can see that we have the lowest correlations very consistently between the extra embryonic neighborhoods compared to those that are forming cell types within the embryo proper. So this kind of gives an approach for looking at similarities in developmental trajectories, and the motivation here is that those in neighborhoods where there's a higher degree of overlap, or a higher similarity, that would sort of dictate where you might want to take a cell type for study in particular conditions in but different biological contexts. Moving forward, what we're doing now is thinking about how we can expand this to more species. So in particular, the macaque and human data. And so how you think about integrating those into this context. So when you're not just interested in pairwise comparisons, how do we think about multi-way comparisons? And in particular, can we do this in a way that borrows information from the phylogeny? We know at the sequence level how related these species are. Can we basically use that as a prior to guide the relatedness between trajectories across the species? I think this is the sort of thing that we're only just now beginning to get enough data to begin to think about doing. But if you want to understand some of these developmental processes, then looking at just pairs of species probably isn't sufficient. But if you begin to now go to multiple and layer on the phylogenetic information on top of it, it's actually got quite a lot of power. So I think that this is something that's going to be quite exciting in the next few years. And I would also say that more generally, these approaches in the context of evo-devo have tremendous power. There are lots of organisms that are evolutionarily much more interesting than the rabbit or the mouse. We use the rabbit and the mouse because they're good lab systems, they're tractable. If you want to understand evolution and development, there are many other organisms that are much, much more interesting than this. Single-cell genomics offers an opportunity to get at them, to really understand their molecular landscape together with genome sequencing techniques. I think in the next decade, we're going to see an awful lot of atlases of development across a wide variety of species, and thinking about computational approaches that will layer the phylogeny together with these developmental trajectories is going to be really important if we want to understand how these work. So I think that this is early, but I think it's going to be very interesting. The other thing that I'm slightly surprised hasn't been done a bit more of is to ask across individual embryos, how similar are developmental pathways? So this is now within a species. So this is kind of work that's motivated more by data generated by Amos Tanai's lab, but they've profiled individual embryos, and from the rabbit and from the mouse. And what you can do there is along any one of these developmental trajectories, say along blood trajectory, you can ask, we can order the embryos individually by their location and then ask, is that order conserved, say in the spinal cord developmental trajectory? So do we have synchronous development across species or across, embryos? So basically, are they equally advanced, or are there different classes where maybe this subset of embryos have developed the mesodermal cell types more than they have endodermal cell types? And can we begin to think about that by looking at embryo heterogeneity within a species? So I think that this sort of early developmental work, especially in a multi-species comparison approach, and even within species comparison approach, there's a lot more to do, and it's really interesting methodologically. Okay, and yes, you can look at these correlations and see that this is now basically looking at the mesoderm trajectory across the rabbit and the mouse, and you can see that it's very highly correlated, whereas the gut tube development is much weaker correlated. So this has got embryonic and extra-embryonic intercalation. So you could kind of see that some processes are more conserved than others. Okay, and then finally, just to close, because I gave this talk whilst half asleep during Simon's birthday symposium. It was the same day that we moved house, and the same day that I formally set up the organization that I was hired to Genentech to do. So it was a bit of a manic day. So I was slightly asleep, I think, when giving this presentation. I thought, see if I could do it more justice today. A final thing to be interested in, maybe not, and you'll all fall asleep again, whatever. What's the best reference sample to use? So we're We've generated lots of atlases. We've generated references in various contexts. How do we use them? We're very used to case control designs, where you have a case, you have a control, you integrate them together, and you look at what's different. How do we now integrate that together with atlases? So we've got these reference atlases in the mouse that I talked about through development, human cell atlas. Are they of any use? So we wanted to explore this a little bit. And basically, the context that we're interested in is we've got a healthy reference tissue, a diseased tissue, and we want to find the disease-associated cell states, so things that are exclusive to the disease. And you could think about doing this in a variety of ways. So we've got these three reference, or these three populations. We've got the disease cohort, and we've got two potential reference populations, the control that's matched for protocol, etc., with the disease sample. And then we've got these larger reference atlases using multiple different protocols, but incorporating, of course, a much larger patient pool. So do we want to just use the atlas? Do we want to use the control? How do we think about combining these different reference data sets? And so basically, so there are simplistically three models that you can think of. The first is we assume we don't have a specific control sample, so we just have the atlas and we use the atlas as a reference. And in this context, what we could do is just project using SEVI, other transfer learning approaches, the disease sample or MNN or whatever, it doesn't really matter. You present the disease cells onto the atlas, and then you ask, are there any particular regions where I have a greater abundance of cells from the disease than the control? And does that tell me something about the novel populations? The alternative is I just use the control samples. So basically I just, you can even create a joint embedding. You don't have to do a mapping here. You just create a joint embedding of the control and the disease samples, and then you could compare them again. But there's also a third way. And as a third way, what you could think about is you've got this atlas. You can now jointly embed the controls and the disease samples into that atlas and then test between them using the locations of the cells within this embedding. What's the advantage of doing this? Well, the atlas might be able to capture rare cell types, trajectories that are not present in your control samples because your control is just too limited in scope. So the thought was that by projecting all of these cells onto the atlas, you would be able to kind of get a better representation of the biology. But by comparing the case and the control, you're also able to control better for technical effects than if you're just to use an atlas, because these control for factors like age, sex, the sequencing protocols that are used. So the hope when we were doing this was that we would kind of get the best of all worlds by using this ACR design. So to test for this, we were talking again with the students about at lunchtime that there seems to be PBMCs are kind of used ubiquitously to test single cell approaches because there's lots and lots of data sets containing PBMCs. So what do? It's that or lung. And in this paper, we did both. But anyway, the PBMCs, so there's lots of data because it's easy to sample from. So in the literature, there are 13 studies, around 1,250 donors. This is a UMAP representation of that full data set. What we did was we considered 12 of these studies as being our atlas. And then we took one study and split it into two, with 13 donors from that study being our pseudo control and 16 being our disease data set. And in the disease data set, basically, we just removed sequentially one population as being absent in the disease relative to the control. Actually, we did the other way around. We added one population here and removed it from this, but it doesn't matter. It's the same thing equivalently. So basically, what we wanted to do is, can we identify the population that's differentially abundant from the disease versus the control using the three reference scenarios that I mentioned before? Okay, so here we go. Right. Okay, so wait a second. It's difficult when you don't have your slides moving. You guys got to scroll. Okay, great. Okay, so these are the different reference designs. So using the atlas as a reference, the control as the reference, and the atlas and the control as the reference. So you can train and learn an embedding. Here we used SEVI using the reference cells, and then use that to map the different query using SC arches onto that reference. So what we can see here is the embedding of the disease samples onto the atlas. And you can see the atlas cells in blue, the disease samples in red. And then this is the out of reference cell population. So this is a population that's present in the disease, but that's absent in the atlas. And you can see that in this embedding here, actually, it's doing a really bad job. These cells from the atlas, from the disease are this population should be absent in the reference. And you can see it's being intermingled really badly. So basically, we have really very little power to identify this, which is bad. We do a bit better when we use the control reference design. So this is now ignoring the atlas, doing a joint embedding of the data sets, and then trying to see whether this out of reference population can be distinguished. And we do an okay job, but actually you can't really see it with the resolution, but there's quite a lot of out of reference cells that are mingled across the rest of the data set as well. Whereas when we look at the combined model where we project the disease and the control data sets onto the reference atlas, you can see that this out of reference population now is quite distinct, and we're able to identify that with a much higher degree of confidence, suggesting that there's a real benefit of using the atlas as well as the controls when we do this. Could visualize this in another way. So of course, we know what's going on here. This works on the bioarchive now. So this is just on the y-axis, plotting the fraction of the out of reference cells that are present in a particular neighborhood. In red are those neighborhoods assigned by Milo as showing differences in abundance between the case and the control, and using the different reference designs. And what you can see in the atlas reference model, when we don't have any controls, you're getting an awful lot of false positives. Similar, though a little bit better, when you use the control versus the reference. So this is just when you embed the control and the disease samples together. But we do best when we project the controls and the disease samples onto the full atlas. We have a much smaller number of false positives, and we seem to have pretty good power to detect the neighborhoods that are out of reference that we really want. So we think that this kind of motivates how you might think of using reference atlases for studying as a way of kind of increasing your power and sensitivity to detect differentially abundant neighborhoods. This is consistent across different out of reference populations. I won't dwell on that here, but across a variety of different metrics, using the atlas in combination with a control and a disease sample, so case control design plus an atlas, seem to give the most power and the best control of the false discoveries. Okay, and then, as I mentioned earlier, we applied this data in a different context. I'm not going to show the lung data, but this is now independent data from a study of COVID-19 where they looked at PBMCs. And so this is now real data. And what we were able to do is to contrast the control reference design, so the classical approach that would work best, where we just have cases and controls, compare that to the ACR model. And what we were able to see when identifying this is we were able to identify neighborhoods that had an interferon signature that's characteristic of severity of infection much more clearly when we're using the ACR design compared to the control reference design. You can see that a little bit better down here. So these are now neighborhoods, and these are neighborhoods where looking at the strength of the interferon signature on the y-axis, and this is against differential abundance fold change between the two conditions, between the COVID and the non-COVID case on the x-axis. And you can see that without the atlas, our ability to detect this signature is much weaker than when embedding all of the data sets into the atlas itself. So this gives us a bit of confidence that what we're finding also has biological relevance. Okay, so that's about it. Oh, I'm just about on time. Good. I think we'd all agree that single-cell genomics have transformed our ability to obtain insights into complex biological processes, and clearly statistical tools will have been and will continue to be essential for enabling these discoveries. I think the cross-species approaches, kind of the multi-omics integration, there's still work to do there. Also, I haven't talked about it today, but beautiful work with Bianca on thinking about spatial modeling of embryogenesis, how we think about this in 4 dimensions. That's all going to be tremendously important. I think we also have to think as a community how we use these atlases. I'm not saying that what we've done is perfect here, but I think that at least in principle, it shows some of the value of something like the human cell atlas, how it could be applied in various contexts. The increased granularity of these atlases can allow biases in rare underrepresented populations and controls to be accounted for. It can allow more accurate mapping of cells from the controlled disease sample onto continuous gradients that again might not be present in references. But also the real value, and as a statistician, it's kind of satisfying that there's still value in controls, of course, controls that account for various confounding factors, sex, age, etc. The combination of these two things is where we're going to get the most power. And in some ways, you might think that this is quite intuitive. It's, I think, still showing it there's value here. And I'm not sure that for me, it wasn't totally intuitive that the atlas would have any value, actually. I thought it was quite possible that a case control model might give you everything you'd want. So I was pleasantly surprised that the atlas actually did seem to really give increased value in this context. So that was nice to see. Okay, the people who make this possible are listed on this slide. This is my current lab. This lab everyone in this slide is still based in Cambridge in the UK. So I'm from California. I have lots of early morning phone conversations with the group, which is interesting, tiring, but still enjoyable. I'm slowly building up the lab in California, postdocs. If you're interested in that, happy to discuss, of course. And more generally, if you're interested in thinking about how any of you could apply your skills in a different context from academia and Genentech, please just reach out to me. Of course, the funders that have made this possible, and everybody in the room for their attention, and Simon for the invitation. Thank you.
Wonderful. So we have time for questions. Are there any in the? Oh, that's good. Okay. Not in the chat. Okay. Any?
Yes, so you highlighted the value of controls in general. Is there any kind of a guideline that you can come up with, or you need X controls or X fractions of the cases in the particular method?
Yeah,
Can you repeat the question?
Oh, sorry. So it's a really good question from Itzik about are there particular properties effectively of the control or the atlas samples that are really necessary and of value? So the answer seemed to be surprisingly not that much. So I thought that if you removed larger data sets from the reference, it would have a much more profound effect from the HCA. It didn't really seem to. I think you need a certain minimum number of cells, and that was, but as long as you have enough data sets in the reference, you're okay. What it did allow you to do was to decrease the number of control samples in a new experiment. So if you used the reference, we downsampled the number of controls, and basically you have very similar results down to having 5 rather than 15 or 16 control samples. So you could basically, by using the atlas, it allows you to cut a bit on the controls because many of the samples are the same. It just the value of the control really is particularly extraneous features related to the sequencing technology or to the biology of the particular disease you're looking at. But by using both, you were able to minimize the number of controls, and that was appealing. So but the actual atlas itself, a slightly smaller atlas seemed to be absolutely fine. So maybe at least in the cell types we looked at, maybe we've saturated the biology, which is quite possible, right? You have 1,000 donors for PBMCs. I think there's probably a limited amount more you can learn by 1,500. Maybe not surprising.
Like for like any contribution I saw was that like they're the atlas without actually releasing the data, and like that way you can map something onto a trained model without actually having access to the whole data set. But here it seems like you're using SRGs in a different way where you increase the power of this test. But then it begs the question, like, why can't you just have all the data together and like optimize the manifold at once? Have you explored like looking at this as a limitation model that you can't do this all at once, and it strongly depends on the order and splits of the data that you feed into the model to get the best like test power?
Yeah, so the question is basically why not just jointly integrate everything rather than map it on? basically, why not treat it? So you could do that. I think the thought was that, or the hope was that individual atlases would be really well annotated, and you would use the labels, or like the cell type labels, to interpret the data as well. So people have spent many years assigning labels or looking at gradients in the atlas, and by jointly integrating it yourself, or integrating everything yourself, yeah, you could transfer labels over to the new data, but you would end up almost inevitably, one way or another, transferring labels. It just depends how you would transfer labels if you jointly integrated, because you probably wouldn't re-annotate the atlas cells, so because they've already got their labels. So I guess the thought was, why don't you just use the atlas manifold rather than create a new one? But we did that in a, so what you could do, yeah, you could do it that way. I guess that it's just one way or another. I think you would transfer the labels over in order to understand what's going on. And as we talked about at lunch, the value and the difficulties of batch effects and things like that might just, but yeah, the order in which you integrate does matter a lot, as we know. And it's kind of, we were talking earlier about how you choose genes. So in another context here, we did a simulation where you removed multiple populations. And that it was in that context where the choice of the highly variable genes really mattered. So that's where some of the models failed quite badly, because they were, once you start removing multiple populations, then you're not able to select the right set of highly variable genes in order to project them jointly together. So it's kind of somehow depends on the number of disease and control samples you have. And if you have too many diseases, then you just don't select the genes that are going to make up the manifold. So it's a different experiment, but it's kind of trying to understand the same thing. When you just, that's really when you have the control reference model. So that's where it failed, because if you have lots of disease and not many controls, then basically what happens is that you, and there is genuine structure in the control data set, but you've just outnumbered it by the number of disease samples. You don't select the genes that are going to give you the gradients and the controls. So that's an interesting imbalance between samples and selection of highly variable genes for then going into downstream analysis. So that wasn't the question, but it's just another point that I thought was quite interesting in the data.
There are a couple of questions on the web, I think.
Oh, do you want me to read them? Okay. Has there been attempts yet to incorporate cell function data into the atlases of your models? It seems that cell functional phenotypes are currently inferred from the gene expression data. Yeah, they are. I think pretty much all single cell studies to date. You might use multiple omics layers, like chromatin accessibility, protein data. The incorporation of functional data generally is, these data sets are more foundational and then motivate the functional experiments rather than the functional data being used. I think as we move forward, more information about morphology from the spatial omics data, information about cell motility, again, from the 4D stuff, that will be powerful to incorporate. I'm not so familiar, maybe people in the room are, where that information has been really directly incorporated yet. I think people have made attempts with morphology and things like seqfish and merfish. So you have as another view, effectively, the morphology, because people have done morphometrics and understood heterogeneity in cells at a spatial level. And you could just imagine that as another view in a multi-view model. I haven't seen that yet. Have I missed something? Where probably I have. Is there a study where people try and do that, where you've got one view is kind of features of cell shape from seqfish and the other matrix is just expression? I have people done that? Did there studies, and did they find anything interesting? No. Okay, so Bianca says people have tried. Pardon? They've tried. Ah, okay. People have tried to do it. So, okay, and then another question. So the question is now about, it's a good question. So it's about the projection of cells from the disease onto the atlas. And would you be better off looking at gene programs rather than cell states? So I think that this is also something that we've, I wouldn't say forgotten, but there's a very cell-specific view of the data, but there's also a gene view where you can ask questions about where is a gene expressed. I know Dana has thought about this, Aviva and I have discussed this on occasion as well. Sort of a fast way of searching where is my gene expressed in the data, where is the particular, which subset of cells show expression of a particular subset of modules, of gene expression modules. We tend to think very much on a cell type level and not on a gene or a gene module level with many representations of the data. There is some interesting work to jointly embed at the gene and the cell level. So you kind of get components that correspond to the cell type and the functional. There's a group at the, Institute Imagine. It's a group in Paris that's done some quite nice work on kind of jointly embedding the expression and the cells in the same UMAP embedding. It hasn't really caught on as far as I can see. I think that some of the interpretability is hard, but I think in the next period, there will be more work again on kind of the equivalent of just where, a really simple question, where is my gene expressed? And how do you define what expression is? And how do you normalize for the total number of genes that are expressed across cell types when I do that? So if I have a smaller number of genes expressed overall and I sequence to the same depth, modulo capture efficiency, do I have to control for that if I set a base level for what is expressed, what is not expressed? So it sounds quite an easy question. It's actually quite a subtle question. And I think one of the reasons for not looking in gene program space instead of cell space is, going back to the question online, is some of these subtleties that you've got to control for differences in the number of genes that are expressed per cell type. You've got to control for the total number of transcripts, ideally per cell type. If capture efficiency, sequencing depth are equal, then that will change some of the expression profiles that you get. And it's quite subtle and difficult to control for that. It's somehow a bit easier to consider a cell space rather than a gene space, but it's a really interesting suggestion and something that's worth following up on.
Okay, I think we've probably used up our allotted time. So thank you very much, everybody. Thank you, John, for a great talk.
Thank you.